and built into the system. Wasn't there a movie? There was a movie about this. The Blue Lagoon. Anybody ever see that horrible movie? Yeah. I don't remember that. I think they figured it out on their own, didn't they, in that movie? So, whether it's something that is learned and needs to be seen, or something that is totally genetic and built into the system, two opposite ways of thinking about human behavior. Two opposite ways of thinking about how we use our muscles to make this relevant to the class. And there is no clear yes or no answer to complex behaviors like that. And you are right, it's probably a mixture of both genes and learning, genes and environment, having to see things. Now, let me conclude this section of the lecture with the important statement again. All of the functions, movement, posture, homeostasis. What's the fourth? Movement, posture, homeostasis. Huh? Skeleton, holding the skeleton together. All of these require the ability of muscles to change their length. So, how do they do it? I mean, this is a really bizarre thing. How do muscles become long, become short? What's going on here? There aren't too many other cells that can do that. This is something very specific to muscles, and to understand how muscles contract, we're going to need to examine muscle structure at the cellular level, the molecular level. Let's look at the cellular structure of muscles, which right there tells you something strange is happening. Okay? Muscle. This is a whole muscle. Each muscle has probably thousands of very long, thin fibers called myofibers. And a myofiber is essentially the cell structure in the muscle. And how do we know that this myofiber is a cell? What is the most important characteristic that makes a structure a cell? How can you tell that something you're looking at under the microscope is really a cell? What does it need to have? It needs to have a nucleus. There are some cells without nuclei, like red blood cells. Remember that last week? Red blood cells don't have nuclei. What else is absolutely essential to defining a structure as a cell? The smallest unit of life. Something that is separated from the external environment and has its own internal environment. What does that? Didn't somebody say it loud? A membrane. What kind of membrane? What do we call that membrane on the outer surface of the cell? The plasma membrane. Plasma membrane is what defines the shape of the cell and totally surrounds the cell. Separating an inside and an outside. So we draw our normal cells as a sphere like that. That's the plasma membrane. That's the boundary of the cell. And it defines in and out. Well, these unusual myofibers are totally surrounded by a plasma membrane. That makes them a cell. They are the smallest unit of life in a muscle. But they have very unusual properties. Unusual properties. RBC cells? First of all, they are very long. Think about how long your muscles are. What's the longest muscle in your body? That's a good question. I don't know. Could be the ones that go up and down your thigh. There's very long muscles that go up and down your back, from the top of the neck to the bottom of your waist. Maybe 3 feet long, there's a muscle. It's made up of cells, myofibers, that are also 3 feet long. There's only one other case where you're going to have cells that are as long as that or longer. And that's going to be in the nervous system. But these are unusual because they're very long. They are also unusual in that they have multiple nuclei. Look at this. Where's one myofiber? It's got a nucleus here, it's got a nucleus here. Here's a myofiber. Look at this. 3 nuclei. Where have you ever seen a cell with multiple nuclei? In your labs or anywhere else? Nowhere. Doesn't happen too often. This is very, very rare. Cells with multiple nuclei are called syncytium. Syncytium. Okay? And they form during development by fusing multiple cells together. Multiple cells aggregate together in these long chains. And each of these cells are still separated by their membranes. And they each have their own nuclei. But at a certain stage in development, these points of contact dissolve. And you are left with a single cell with multiple nuclei. And another interesting characteristic of these myofibers, if you look at this carefully, they each make contact with the nervous system. There is, I should write it here, please see, each single cell in the muscle, each myofiber contacts a nerve cell. And as we will see later, this is absolutely essential for the functioning of the muscle because it is signals from the nerve to the muscle that tells the muscle how much to contract or whether to be totally relaxed or what. It all depends on signaling from the nerve. Now, the amount of contraction in the whole muscle in the whole muscle is going to depend upon the contraction of individual cells, of individual myofibers. You can have a contraction where some myofibers of the muscle are contracted and some are not. You can have a contraction of a muscle where the amount depends on the total amount of contraction of each one of those myofibers. So essentially what this means is that the property of the whole muscle depends upon the contraction of individual cells in the muscle. They can add up together. Okay, now how are we going to understand how these cells do this strange thing? How do they change in length? How does that happen? Well, what do you do when you want to understand something, how a cell behaves? What do? What do you do in lab? You look at it. You look at it with what? Participation. Huh? This is a song. This song, that's anticipation. You know the song Anticipation? No, you don't know the song Anticipation? No, I'm going to change it to participation. All right. You look at it with a microscope. You look at it with a light microscope. And what do you see? You've done this. groove here, okay? You look at a muscle in the light microscope, and what do you see? You see stripes. That's one of the reasons these skeletal muscles are sometimes called, they actually have another name. Skeletal muscles are sometimes called striated muscles. Striated means striped. Okay, here you are, an explorer in this new land of seeing something in a muscle. Imagine that nobody else has ever seen. You have the liberty to give names to structures. Describe the structures that you want to distinguish as being unique to the skeletal muscle. What are they going to be? Well, we can be real original and say, whoa, I see dark stripes. And I'm going to call those dark stripes A-bands. You can be even more original and say, well, a real distinct characteristic are these light stripes. And I'm going to call those light stripes I-band. You're the first one to see those. You get to name whatever you want to name them. I don't know who to call them A-bands or I-bands or why. It's the name they got. Now, if you've got really good eyes, you can see a thin line going down right to the middle of each of those I-bands. Can you see it? No? Zoom in. You change the objectives in our microscope. Try a high about so it gets too pixely. Can you see it now? Hints of a thin, darker band running right down the middle of the I-band. Whoa, that's an interesting structure. What do you want to call it? Look at your notes. What do you want to call it? Hey, that's a good name. Let's call that a Z-disc. Okay? Did you all see it? Did you all see it? Now, we are going to, since we are the first discoverers in this new land of the muscle under the light microscope, we're going to define another structure. We're going to define a structure called a sarcomere. Okay? And we're going to say this sarcomere is the distance between two Z-discs. Okay, so zoom in. There's a Z-disc, and there's a Z-disc, and the distance between them is a sarcomere. And that means also that there are probably thousands of sarcomeres along the length of the cell, along the length of the myofiber. Thousands of them. Now, what if some of these structures that we see, the sarcomere, the A-band, and the I-band, are in some way involved in producing contraction of the muscle? How are we going to test that? Well, we're going to look at muscles at various states of contraction, and we can look at it very simply at the level of the light microscope. Here is a graphic drawing. Okay, we're getting away from the real image in the microscope, but many people have seen those pictures in the light microscope and they draw them like this. Here is a totally relaxed muscle. Here is a totally contracted muscle. What changes are occurring in those structures that we saw under the light microscope when the muscle contracts? And does this indicate that perhaps these structures are involved in producing the contraction? Well, look at this. Z to Z. Here's a sarcomere. In its relaxed state, the sarcomere is long. In the contracted state, the sarcomere is short. There is a change in length of the sarcomere as the muscle contracts. What about the A-band? Is the A-band changing in length as the muscle contracts? No, it's staying the same. whether it's fully relaxed or fully contracted, A band has the same length. What about the I band? I band decreases in length as the muscle contracts. So there are changes in these structures that we saw with the light microscope that occur when the muscle contracts. So we can say muscle contracts by the change in length of the sarcomere. Does that help you any? Does that give you more understanding of how a muscle contracts? No! It's just descriptive. We're just using different words for the same thing. Decrease in length of the sarcomere, decrease in length of the muscle. Come on, we want to know what happens when the sarcomere changes in length. What's producing that? We've pushed the limits of the light microscope. We need to examine the muscle at a higher magnification. Light microscopes can magnify things about 1,000 times maximum. But if we use an electron microscope, you can magnify things at least 100,000 times. You can see things with an electron microscope that you can't see with a light microscope. So to try to improve our understanding of what happens when the sarcomere changes in length, we're going to look at the muscle in the electron microscope and see what we see. And let's go exploring in this new structure that was revealed to us. What do you see here? What are the A band and the I band made out of? What we see here are two types of filament. And a filament is a long, thin, thread-like structure. Okay? And you can see that the A band has thick filaments, as seen here, darker. The A band, or there are thick filaments, let's put it this way, thick filaments that run the length of the A band. There are, on the other hand, in the I band, thin filaments. I band has only thin filaments. But in the A band, there is an overlap of the thick filaments, and in these portions, thin filaments. So the A band actually has both thick and thin filaments. Okay? So this is what those dark structures, those A bands are. They're thick filaments, fibers that run across. There are, I band is made up of thin filaments. If we take a cut right through this A band, we see that there is like a crystal structure. Darker circles, bigger circles are the thick filaments, surrounded by the thin filaments. What are these thick and thin filaments doing to produce contraction? How are we going to determine this? I'm not going to let you go until we answer this one question. How are we going to determine what the roles of these thick and thin filaments are in contraction? We're going to do exactly what we did with the light microscope, which is, what do you think?
Look at it.
What are we going to do? We're going to look at contracted muscle, and we're going to look at relaxed muscle and muscle in between. We're going to look at them in the electron microscope and see what happens to these thick and thin filaments.